The study focuses on how invading gliomas, a specifically type of fast-growing brain tumor, affect the perivascular space. Astrocytic infeet provide immense function in the brain, covering approximately 99% of the cerebrovascular surface. Astrocytic infeet serve as exchange sites for ions and metabolites, maintain the tight junctions that serve as the blood-brain barrier, and regulate vascular tone. These processes have been termed neurovascular coupling (NVC) as the infeet adjusts local cerebral blood flow to match regional neuronal energy demand. Due to the importance of astrocytic vascular coupling, when the space that these astrocytes occupy is impeded upon, there are vast consequences. Gliomas, a common invading presence in the brain, are extremely fast-growing, mostly malignant tumors. These tumors often occupy the perivascular space and co-opt vessels as satellite tumors, which are lesions within proximity to the original tumor that are considered intralymphatic extensions of the primary mass. The study of focus utilized mouse models of gliomas in order to investigate the tumor's effect on astrocytic infeet and therefore the functions that the infeet typically provide. In this study, the interaction of migrating glioma cells with pre-existing vasculature was investigated in order to gain understanding and insight for treatment. To complete the study, mice were bred and maintained in a specific pathogen-free facility. These mice were housed in groups of 5 and subjected to a standard 12-hour light, 12-hour dark cycle. The mice were either implanted with GI261 DS red mouse gliomas, D54 EGFP human glioma cells, patient-derived xenograft tumors, or non-malignant glio cells. These tumors were surgically implanted into 6 to 12-week-old immunodeficient mice of both sexes. On the day of surgery, once animals were anesthetized by 2 to 5% isoflurane, a midline scalp incision was created. A 0.5 millimeter burr hole was made at 1.0 to 2.0 millimeters and 0.5 to 1.0 millimeters posterior from the bregma. These tumors were allowed to grow for 2 to 4 weeks while the mice were monitored. Body weight was monitored in order to determine which mice indicated tumor growth, and those with the most significant weight loss were chosen for experiments. For analysis of effect of gliomas, immunohistochemistry was utilized, and images were taken using laser scanning and microscopes and electron microscopy. Furthermore, in vivo imaging was utilized, in which the skull was removed and arteries and arterioles were observed. The effect of gliomas on the blood-brain barrier was analyzed through permeability assays analyzed through laser scanning microscope. Investigation: the extent to which glioma cells associate with blood vessels. Investigation into how glioma cells displace astrocyte infeet from the vasculature. An investigation into how perivascular glioma cells assume control of vascular tone. Gliomas were generated using D54 EGFP implanted into the cortex of the immunodeficient mice, and they proliferated for 2 to 4 weeks. The D54 and GBM22 gliomas form loose tumor borders within the cells while also invading the parenchyma using the blood vessels as a scaffold. Most of the D54 glioma cells associated with the capillaries took up 47.5% of the total vessel area. Since only a small percentage, 1.6% of large vessels and veins were targeted by the gliomas, it was concluded that there was a preference of capillaries over arterioles and venules, answering the first question of the research. The next question raised was if gliomas target venules filled with venule or arterial blood. 54.46% of the gliomal covering was over venules or veins, and 36.38% over arteries greater than 10 micrometers. 2-photon imaging was also utilized to show the direction of blood flow and size of the vessels. Most of the gliomas associated with the blood vessels throughout the entire vascular tree, with a clear preference for small capillaries. Immunofluorescence of CD31, PCAM, aquaporin-4, AQ4, and EGF5 expressing human glioma cells, D54, implanted in the cerebrum of immunodeficient mice is seen in this figure. It is clear through this imagery that there is overlapping and subsequent displacement of the astrocyte infeed by tumor cells from the vessels. This interruption could considered normal interaction through the previously recorded mechanism of retraction due to the involvement of aquaporin-4. This is where the EGFP expressing human glioma cells become relevant, expressing in red in the figure. It was observed that glioma cells indeed displace infeed away from endothelial cells, separating from normal pathology. At this point in the study, the specific displacement was further researched. Of importance in these figures is the darker appearance of the glioma cytoplasm, and therefore the rougher ER and the high nucleus to cytoplasm ratio. These interesting criteria were used to identify the placement of the glioma cells. It is seen that glioma cells can gain direct access to the abluminal side of cerebral vessels through the displacement of astrocytic and feet. However, this was not unilateral, as some in feet were intact. This gives evidence for the original hypothesis that gliomas interfere with the blood-brain barrier. Vessel cooption by glioma cells breached the blood-brain barrier, concluded from the presence of the TRITC albumin and the vasculature even at distant sites in the parenchyma and the tumor-bearing vessels. This information was validated as glioma pathology as no leakage was observed in the other cortical glioma-free regions. This occurred through the glioma's access on the abluminal side of the cerebral vessels. It was found that activation of astrocytic GCPRs caused an increase in calcium and activation of phospholipase A2, which then converts membrane phospholipids into AA. AA can be released from the astrocyte and converted to 20-HETE, causing vasoconstriction or metabolized into PGE2, which dilates vessels. Single astrocytes surrounding vessels were stimulated through pharmacological methods, and the response was either constriction or dilation, which occurred in a matter of seconds. Calcium is a cellular signal that stimulates a vessel diameter change and type of response, dilation or constriction. In agreement with previous studies, application of TACPD caused vasodilation or vasoconstriction in tumor-free vessels. Similarly, norepinephrine application induced constriction or dilation in glioma-free arterioles. In contrast, arterioles encased by glioma cells showed a significant decrease in vessel response after TACPD. This indicated that the glioma cells had an impact on vascular tone. Also investigating vascular tone manipulation, calcium engaging the astrocytes close to the vessels associated with D54 glioma cells did not respond, meaning that astrocyte-derived signals do not reach the vasculature. The constriction and dilation of vessel is complementary to current studies, however, no apparent new conclusion was able to be made. It was shown that a majority of human glioma cells that traveled outside of the main glioma mass associated with newly generated blood vessels at different stages of the disease and inserted themselves between the infeed and the endothelial wall, confirming previous reports using rodent glioma cell lines. This was expanded upon to determine the nature of this interaction and the effects of the interaction. The study concluded that gliomas preferentially associated with small calibre capillaries, resulting in clear displacement astrocytic infeed from endothelial or vascular smooth muscle cells and restriction in movement of the infeed. This causes a focal breach in the blood-brain barrier, bringing up immense pathogenic implication. In normal pathology, constant maintenance of the endothelial cells is required by the astrocytes. With the glioma, however, the connection between the two is interrupted, and therefore so is the maintenance, allowing for the breach seen in the study. The most clinically relevant result from the study was that it found that single glioma cells are sufficient to disrupt the blood-brain barrier. As just a few cells were sufficient to cause albumin leakage. This was of particular importance because of the implications that arise from this finding. This impacts the course of chemotherapy typically recommended depending on the stage of the progression of the tumor. The article contradicts current treatment progression, proving that small early-stage gliomas are susceptible to chemo. This portion of the research is one that could be focused on for further research, delving into the implications regarding blood flow in the tumor-associated brain and the ability to locally deliver chemotherapeutic drugs in disease. Furthermore, astrocyte-mediated gliovascular coupling was determined to be as lost. As the glioma cells ceased control over the regulation of vascular tone, the vascular tone was concluded to be regulated by the glial cells through calcium-dependent release of potassium. Further studies could further examine whether parasites contribute to blood-brain barrier breakdown in response to glioma evasion. Another further study would include disrupting the tumor control over the blood flow in order to determine if impairing or invade the growth should be exploited therapeutically. The expression of the glioma cells could be altered to see if the target diameter of the vasculature changes.